Well, I've previously been focused on what happens before the modelling stage in the data science lifecycle. This week, we're going to transition our focus to what happens afterwards. We're going to have a look, once we've fitted our models, how we can get those into production, and we're going to begin by focusing on two key properties that will be required. Namely, that's going to be whether our model can be reproduced and whether our results can be replicated. So, as well as focusing on the first part of the data science cycle, we've also been very much focused on the first part of the name. We've been really keying in on the data aspect of data science. What we're going to think about in this video is more the other side, the science aspect of data science. So, as data scientists, we like to think that we're applying the scientific method in our work. As scientists, we start with a question that we want to answer or a problem that we want to solve, and then we turn to the existing literature. We want to establish if this is a well-known problem that lots of people have solved before, and if it is, that's fantastic. We can learn from their efforts. If it isn't, then we proceed to gather our own evidence and combine this with whatever existing knowledge we could scrape together. Finally, we draw conclusions from this synthesized information. When we're doing this, we acknowledge that the conclusions that we're going to reach aren't actually the truth. They're just our current best approximation of it. What we've created is a usefully simplified model of the messy reality that we live in. We'll happily update this model as we become aware of new evidence, whether that new information supports or contradicts our current way of thinking. This sounds like a great approach, and it is. In an ideal world, this is how both data science and science as a whole would progress. However, just like our models themselves, this is a simplified and, in this case, an idealized description of what really happens. The aims of a data science project are rarely framed as a clear and unambiguous hypothesis, for which we design a single study and perform a single statistical analysis. Apart from in really special cases like A/B testing, we have a much more general aim for our data science projects. One example of this is that we might want to construct a model for a given phenomenon, and we'll try many variants of that model along the way. By taking this more relaxed approach to data analysis and modelling, data scientists can run the risk of finding spurious relationships within our data that don't hold more generally. If you're trying to find a relationship between a quantity that you're trying to predict and lots of samples that are just entirely random noise unrelated to the outcome, eventually you're going to find a relationship that's significant, even though there's no true underlying relationship there. While it's not really quite this extreme, we can lead ourselves into believing that our findings are much more significant than they actually are. And that's exactly what Gelman and Lopen are talking about in this paper about the Garden of Forking Paths, which is on the reading list for this week. Okay, so our methods for investigation as a data scientist might not be entirely in line with the scientific method, but that should be balanced out by the results of other studies that exist when we combine this information, right? Well, the second worrisome aspect of this process is that we can't always trust the published literature to be a fair representation of everything that people have tried in the past. Studies that don't provide strong evidence against the null hypothesis that they're testing rarely make it into publications, reports, or the news, and this is largely because of the way that scientific inquiry is rewarded, whether that's in the academy, in business, or in the media. Generally, funding and attention end up going to studies with new findings rather than those which aim to confirm or strengthen the findings of existing results. This systematic problem incentivizes scientists to do things like massaging the numbers to obtain a p-value that's just less than 0.05, so that a result can be reported as statistically significant. This process is known as p-hacking and can occur through deliberate malpractice, but more often it's a result of scientists not generally receiving adequate training in stats. As statistically trained data scientists, we know that a declaration of significance is no indication that the effect that we've discovered is meaningful, and that this 5% significance level is entirely arbitrary. However, we do need to be aware that this isn't the case across all of science, and even we aren't immune to the societal and systematic influences that favour the publication of novel results over confirmatory ones. We can really see the effect of this arbitrary 5% cutoff in this plot of the distribution of p-values in all medical publications. There's a clear step change between the large number of published studies with p-values less than 0.05 and the much lower number with p-values greater than that. These same systematic influences also lead to a much more subtle problem than this direct p-hacking. As an example of this, consider a model that has an obvious but truly unnecessary additional property that might be added. So, for example, here you might think about adding a completely unneeded term to a regression. Because this extra term is really obvious as an extension, it presents itself as low-hanging fruit to researchers. This means that many scientists are likely to independently design experiments to test out whether or not adding in that extra term is going to provide some sort of improvement. The vast majority of experiments are going to provide insufficient evidence against the null hypothesis and then don't get developed into published papers or technical reports because they're just confirming the status quo. However, after enough people have attempted this, some scientist will eventually get lucky and find a significant and potentially relevant benefit, but where one truly doesn't exist. It's just a spurious finding. And then of all of these experiments that were done, it's potentially only this one with the spurious result that makes it onto the public record. All these studies being left in the proverbial desk drawer induce something known as publication bias in the scientific literature. When we come to assess the state of existing knowledge, this means that we're unable to properly assess the importance of findings because we lack the context of all those null results that went unreported. This exact same process means that the results of many more scientific studies than we might expect can't be recreated when tested in independent replication studies. Okay, so let's focus in now from the general scientific view of reproducibility and replicability into what this means for us putting our data science work into production. Let's start with this idea of reproducibility. This requires us to be able to recover the exact same numerical summaries as the original investigator did. In particular, this means that we should be able to reproduce the exact same point estimates and measures of uncertainties, which then means we'll draw the same conclusions as that original investigator. When we're putting our work into production, there's several reasons why we might require it to be reproducible. The first is a logistical one. Production code needs to be robust and it needs to be efficient. This means that your code will often be refactored, rewritten, or translated into an entirely different language before it's released to the world. If the results of your original study aren't reproducible, then there's no way to verify that this translation has been done correctly. The second point is that if a problem is identified in your work, say it is put into production, and then a customer raises a complaint that their loan application was incorrectly rejected, you need to be able to go back and recreate that exact instance to diagnose if there's a problem and exactly what might have caused it. This is an important thing to note here, that just because findings are reproducible, that by no means implies that they're correct. We could have a very well documented but highly flawed analysis that is entirely reproducible, but is also completely unsuitable or just plain wrong. In our data science projects, we've already taken several steps that greatly improve the reproducibility of our work. Although we scripted, tested, and documented our work to improve the management of our project, these exact same decisions improved the scientific quality of our work too. This puts us in a strong position relative to the scientific literature as a whole. We're at a point now where it's almost standard to publish data along with papers, but for a long time that wasn't the case. It used to be that if data were available at all, this might only be by request. Nowadays, we're in a similar situation when it comes to code. It's still far from standard for the analysis code to be required, to be put up to detailed scrutiny as part of the peer review process. This might seem unreasonable, but with a little bit more context, it's not so strange. Across many scientific disciplines, coding-based approaches to analysis aren't the standard. In these disciplines, it's often the case that statistical software with a graphical user interface is used instead. The idea here is to lower the barriers to access, to allow scientists to analyze their own data by using tools that have a combination of menus and buttons rather than requiring coding skills. However, the downside of this is that these interfaces often leave no record of how the data were manipulated, and the software itself can be highly specialized or proprietary, being incredibly expensive to use. This combination means that when full datasets are provided, that doesn't necessarily mean that other people can reproduce the original analysis. I should note here that none of this is meant to scold or disparage scientists who use this type of software to allow them to perform statistical analyses. We're well aware of how much time it takes and how much effort is required to learn how to use and implement statistical methods correctly. This is the exact same time and effort that other scientists invest in learning their subject matter, so they can get to the point of doing interesting research in the first place. I think this is one of the wonders of data science. It gives you the ability to work in multidisciplinary teams where individual members have different specializations and bring their own strengths to the project. This is also a good time to pause and check ourselves, because we can easily befall the same fate. Yes, it takes time to learn the skills and practices to ensure reproducibility, but it also takes time to implement them, and the time of an expert data practitioner doesn't come cheaply. If you wait until the end of your project before you start thinking about making it reproducible, then you're likely going to be too late. The time or the money will have run out. So moving on to replicability then. Statisticians are well aware that if we were to repeat an experiment, we'd get slightly different data. This in turn would lead to slightly different estimates and slightly different results. Ultimately, this is the core problem that statisticians get paid to worry about. We want to assess whether those changes will be small enough that the substantive conclusions of the study are not impacted. Okay, yes, the point estimates are going to vary slightly, but do your conclusions about the existence, the direction, and the magnitude of an effect still hold? Alternatively, if you're estimating a relationship between two variables, is that same functional form chosen when the study is repeated? A general scientific definition of replication takes this idea in a bit broader of a sense. It asks whether the key properties of your results could be replicated by another person. In the context of getting your work put into production, what we care about is whether your results, your key findings, will also hold when applied to future instances from the production population that might differ in some ways from those that you've seen already. Since we're approaching data science from a statistical viewpoint, then we're well accustomed to these sorts of considerations. Whenever you perform a hypothesis test or compare two models, you're actively taking steps to ensure that that comparison isn't only valid for the particular sample, but is also true out of sample. This idea is the whole reason why data scientists make training and test sets in the first place, as an approximate way to test for this sort of ability to generalize. Whenever we do any of these things, we're asking ourselves, will this good performance replicate if we had different data, different inputs? Of course, it's important to acknowledge that this training test split or bootstrap resampling or asymptotic argument can only ever approximate the ways in which our sample differs from the population to which our production model will be applied. The only way to truly assess this out-of-sample performance and our model's ability to generalize is to put that work into production. The concern here is that putting this work into production opens us up to risk. What if our findings don't generalize, and our new model is actually much worse than the current one? Well, it's not possible to both deploy our new model and also avoid this risk entirely. However, what we can do is take some small steps to mitigate our level of exposure to that risk. In the most risk-adverse settings, we might implement something known as a shadow deployment of our new model. In this setup, the current model is still used for all of the decision-making, but our candidate model is also run in the background, so we can see how it might behave when released into the wild. This is good in that it means that we can identify any points of catastrophic failure for the new model, But it's also really expensive to run two models in parallel like this, and it can give us only very limited information about our new model. So let's focus, for example, on our model being a recommender system on a retail website. A shadow deployment will let us check that the new system functions correctly and that we can gather data on what products it recommends to each customer as they come along. And we might investigate how these recommended products differ from those that were recommended by the current system, the products that the customer actually sees. The problem is that this shadow deployment can't tell us what the customer would have done in this case, had they been shown the new set of products instead. This means that a shadow deployment can't answer questions like whether the new system leads to equivalent or greater revenue than the current system does. To answer that type of question, we need to do something called a parallel deployment. Parallel deployments are related to A/B tests, where both have the current and proposed new model running at the same time. This allows us to truly test whether our findings generalize to the production population because they're being exposed to the new model, and it also allows us to control our level of risk exposure by setting the proportion of times each model is shown to the users. The more instances that we assign to the new model, the faster we're going to learn about its performance, but also the greater risk that we're putting ourselves at if that model is in fact inferior. So in data science, we rely a lot on the use of stochastic methods and models. These are often used to increase the chance of our findings being replicated by another person or when they get put into production, which is great, but they also make it much more difficult to ensure that our exact results can be reproduced, whether that's by another person or our future selves. So let's take a look at some of the key sticking points for reproduction and replication in statistical applications. The first sticking point are Monte Carlo methods, which are any modelling, estimation, or approximation technique that leverages randomness in some way. We've seen examples of this already to improve the probability of a successful replication, with the most obvious example of this being the random allocation of data in a training or test split. A second example that focuses on improving replication is to use bootstrap resampling to approximate the sampling distribution of a test statistic where this isn't known. This might be a parametric bootstrap method, where alternative versions of our dataset are generated by sampling values from the fitted model. Alternatively, it might be a non-parametric bootstrap, where we generate these alternative datasets by sampling from the original data with replacement instead. These Monte Carlo methods can also be used to express uncertainties more generally, or to approximate difficult integrals. These are both common applications of the Monte Carlo method during Bayesian modelling, where unless our models are particularly simple, the posterior or posterior predictive distribution has to be approximated by a collection of values sampled from that distribution. What's common to all of these methods is that every time we run them, we're going to get slightly different outcomes because they rely on random sampling. For our work to be replicable, we need to quantify this level of variation that we'd expect. So for example, we might be trying to estimate the posterior mean. We could do that by taking a collection of samples from the posterior distribution and calculating their mean. But if we had a different sample from that posterior distribution, how much would we expect that estimate to change by? Sometimes, as in this case, we can appeal to the law of large numbers to help us out. So if we take more samples, then the variation between these different realizations is going to be reduced. If we can collect enough samples, then our estimated mean can be made stable up to our desired number of significant figures, but that might potentially take a very, very large number of samples that are just too costly to collect. Setting our number of samples appropriately here can help us to ensure the replicability of our findings, but to make our results more reproducible, we have to ensure that we can exactly reproduce those remaining unstable digits in our estimate for any particular realization or sample from the posterior distribution. The main way we can do this is by setting the seed of our random number generator, which is an idea that we'll return to shortly. The second sticking point that often causes challenges when making reproducible and replicable data science projects is optimization. If an optimization routine is used for parameter estimation, then we have to ensure that the results that they find for a particular set of data are reproducible given that optimization configuration. So to be clear here, that configuration might consist of a particular dataset, an initial set of starting parameters for the optimization routine, and an initial step size. And this would be accompanied by a learning rate, which controls how that step size changes, and the maximum number of iterations that we're going to perform as we seek to find the optimum value of this function. To ensure that our results are reproducible, then we're going to have to make sure our optimum is stable across all of these hyperparameters. We additionally need to be concerned about replication here, particularly in the case where our optimization incorporates some form of stochasticity. For example, this might be a stochastic gradient descent or simulated annealing, where the search procedure is itself random in some way. We then need to be careful that our optimization, even when it started from the same point, can recover the same final value at the end of our final iteration. So returning then to this idea of random numbers and seeds, it's often the case that we have stochastic elements within our work that we can't use brute force to eliminate. Maybe this is beyond our computational abilities, like I was saying before, or maybe the number of realizations is actually an important fixed aspect of our study, and we can't grow that to reduce the variability. Fortunately, this isn't too much of a problem, because in computing, it's very unusual to use truly random numbers. Instead, what we have is a complex but deterministic function that generates a sequence of numbers that are statistically indistinguishable from a series of independent random variates. We've got a deterministic function that makes something that looks like it's random. This function works by generating the next term in the sequence of pseudo-random numbers based on the value of the current number. This means that by appropriately setting the starting point, we can always get the same sequence of pseudo-random variates being generated every time we run our code. In R, we set this starting point using a function called set.seed. This is especially useful for simulations that involve random variables because it allows us to create the same results exactly every time we use our code. This not only makes it possible for others to recreate our results exactly, but can make it a lot easier to test and debug our own code. There are a couple of things to be aware of when you're using seeds to control the randomness within your simulations. When you're running code sequentially and interactively, then setting a seed is going to be about all you're going to need to resolve reproducibility problems. However, you really should take care when combining this need for reproducibility with some of the methods we'll see for speeding up your code. In some cases, the strategies to optimize your code's performance can interfere with the generation of random numbers and lead to some really quite unintended results. The first case to be aware of is that when you're writing code that's executed in parallel across multiple cores or processes at the same time, then you have to really carefully consider whether or not to give each of these processes the same seed value. The correct decision here is going to be context specific. It's going to depend on the interpretation of the random variates that you're generating. If you're making a comparison between two different processes, then it might be important that the random aspects are kept as similar as possible. While if you're using parallelization only to gain speed in your code, then that might not be such an important thing at all. The final thing I want you to be aware of is that you can run into these sort of issues with pseudo-random number generation when interfacing R with other programming languages. So for example, when you're writing an R wrapper for C++ code. Since R was developed as a statistical programming language, it's actually a lot better than most other languages at dealing with random numbers. Other languages might use slightly different algorithms for generating these random numbers, and the quality of those generated numbers can vary. The one thing that's really quite important to make sure is that the seeds that you're setting are appropriately passed between those languages to ensure that everything stays in sync and the correct sequence of random numbers is generated. So let's wrap up this section here then. To get our work put into production, it should be both reproducible and replicable. This will ensure that we can recreate the same results that we had in the past from the code and data that we have available to us. It'll also mean that the core results that we've found in our modelling sample are more likely to hold when we apply that model to our production population. We've pointed out that randomness is a key part of most data science workflows, that this can lead to some real reproducibility nightmares. We can manage these by appealing to the stability of averages in large samples, and by explicitly setting the sequence of pseudo-random numbers that we generate. Finally, I took a little bit of time to warn you of some important considerations when you combine efficiency with replicable workflows. With the ideas in this lecture, you can now go and use good data to do good science. Thank you very much for watching.